In this video, we will discuss about in some more depth about this prominent role of macrophages as effector cells in the T cell based therapy models. So in the previous video, we have understood that they have made a point that macrophages also plays a role along with the activated T cells to kill the tumors. So in this video, we will discuss some more evidence that are presented in the paper that supports this point. So in order to check the role of different effector cells in the T cell based therapy models, what they did is they use antibodies to deplete different immune cells, such as in order to deplete CD8 plus T cells, they use anti CD8 plus T cells or anti NK 1.1, and in order to deplete neutrophils, they use anti LY6G, and in order to deplete macrophages, they use anti CD115. And this is the experimental models that they have used. So for this RMA Q1B negative engraftment mice model, they use these different antibodies at different time intervals and the vaccines at 11th day. And for this different mice model, that is B16F10 engraftment tumor mice models, they use these vaccinations at 3 days, 10 days, and then by specific antibodies injection at 12 days, 15 days. And this depleting immune cells antibodies are used at different time intervals that are shown in this figure. And thereafter they check this tumor burden at different time intervals after the injections of different vaccines and different antibodies against different immune factors. And what they find out is that for the first model, that is RMA QA negative engraftment mice models, they have find out is that when they use this anti CD8 plus T cells, they do not see any increase in the survival rate. However, when they use this anti NK 1.1, what they have find out, they have find out that there is a increase in the tumor burden at a higher number of days, or we can say that the tumor suppressions are high when they use this anti NK 1.1, but they are low when they use the they do not see any change when they use these vaccines and anti CD8 plus cells. So we can see that at this time the results of this anti CD8 plus cells is similar to what they have observed for the untreated cells. And when they check the other results for this BF16F10, what they have find out is that they do not see any they do not see any they see that complete reductions in the tumor burden with this anti NK 1.1 cells. So right now we see that when the NK 1.1 has been depleted, they see improve in the survival rate. Now they in the panel E and F, they shows that this anti immune factor antibodies works well. So for the CD8 plus cells, since they use anti CD8 plus cells, they can see that when they use anti CD8 plus cells, there is a decrease in the CD8 plus cells populations, and similar results when they use the anti NK 1.1 cells. And for this CD45 plus cells, they also see the same results. That is, when they use anti CD115, they see a reduction in the macrophages, and when they use this Ly6G, they see reduction in the neutrophils. So far, what they have find out is that when they suppress this neutrophils, they see an increase in the survival rate. and along with that, what they have also find out in this panel G is that with a tumor, what they have find out is that with the reduction in either CD8 plus cells or the macrophages, or both of them, they they see a decrease in the survival rate. That can be seen here, that when they use this anti-CD115, that is macrophages, they see a change compared to the untreated cells. And however, when they use anti-LY6G, they also see a change in the tumor burden. But when they use both the combines antibodies, that is anti-CD11 and anti-LY6G or anti neutrophils, they see a different changes that can be seen here. That is, the survival rate goes down compared to the case where individual antibodies are used. And these things they have seen, similar things they have seen in this B16F10, where they have seen that when they use this anti-CD115, they see a reductions in the tumor burden. But when they use this anti-LY6G or anti-neutrophils, they do not see a decrease in the tumor burden. And let me re-explain this panel G. Here we can see that when they use only the vaccine, there is a tumor decrease, but to some extent. And when they use this anti-LY6G or anti-neutrophils, neutrophil depletion antibodies, they see slightly more reductions in the tumors. And when they use this CD115, they see a more anti-CD115, they see more reductions in the tumors. But when they use both of them, they see a further reductions in the tumor burden. So this indicates that this both of them, that is anti-CD115 or anti-LY6, plays a role towards decrease in the tumor or increase in the survival rate. And same things we have seen here. Here mainly the results are being shown for are being reflected for this anti-CD115, but anti-LY6G has no effect. That is, neutrophil has minimum impact on this B16F10 based mice models. And when they check the impact of one antibody's depletion on the other antibodies, what they have find out is that when they deplete this anti-CD115, they see that neutrophils are also decreases. Neutrophils are also they see that the macrophages are decreased, but they do not see much decrease in the neutrophils. But when they check the neutrophils amount, what they have find out is that let me re-explain this. For the macrophages, when they use this anti-CD115, they see a decrease in the macrophages, but but when they use this LY6G, they do not see a decrease in the macrophages uh, in this case. But when they check the neutrophils, what they have find out, when they use the anti CD115, they do not see a decrease in the neutrophils, but when they use and when they use this LY6G, anti-LY6G, in this case also they do not see too much decrease. So overall, there is not much change in the neutrophils that has been observed, whereas when they use the when they check the macrophage amount, this macrophage amount in the tumor macro environment. So these are about panel I and J are about the compositions of the immune cells at the tumor macro environment. So those immune cells at tumor macro environment decreased at the macrophages, but not at the neutrophils. but a neutrophils amount does not get much impacted. So that can be seen from this panel I. And for the panel J, what we can see about the percentage of the macrophages, so which macrophages got decreased. And what they find out is that this M1 macrophages got decreased, whereas there is not much change has been observed for the M2 type macrophages. It indicates that M1 macrophages plays a role along with the activated T cells towards tumor suppression. And when they check the results for the B16-1F, in this case also they see that uh, overall macrophages goes down in the total percentage of live cells, and when they check the M1 and M2 macrophages, they see that M2 macrophages goes down compared to the vaccine and bispecific antibodies case. So this indicates that these macrophages are being recruited by the somehow been recruited by the activated T cells. So that point is not made here, but immune cells amount is decreased. When the immune cells amount is decreased in the tumor microenvironment, the efficiency of the T cell based therapies models goes down. So the reason why this T cell therapies does not work in some cases is not because the T cells is not performing well, but the amount of macrophages might be low or might not be able to reach to the site of tumors. So this is how they made a point that macrophages plays an important role as effector cells in the T cell based therapies, and especially M1 type macrophages, which has anti-tumorigenic properties, generally anti-tumorigenic properties. So I hope you understood these experiments and their results and the final point that they have come from this figure. If you want to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.